So hello, my name is Stephen Griffin from the University of Leeds, and today I'll be telling you about our AMMF-funded PhD project investigating the way in which hepatitis C virus infection is capable of driving intrahepatic cholangiocarcinomas. And I just wanted to start the talk really by acknowledging the people that have really been doing the majority of this work. And the first person to highlight is Alexandria Kilvington, who's our current PhD student funded by AMMF, and she's been doing all of the cholangiocarcinoma aspects of this, which I'll tell you about shortly. But it was also following on work from two MRC funded members of staff, a PhD student, Abigail Bloy, and Dr. Matthew Bentham. So why look at virus-driven liver cancers, primary liver cancers? Well, we know that viruses such as hepatitis C and hepatitis B are responsible for the vast majority of hepatocellular carcinomas, that is, primary liver cancers arising in hepatocytes within the liver. But they're also very much associated with cholangiocarcinomas, specifically those originating in the intrahepatic portal part of the portal tract. And we know that the cells that line this area of the bile duct are different compared to the extrahepatic milieu. And we also know, interestingly, that when these cancers are associated with hepatitis virus infection, there are a great many similarities between the cholangiocarcinomas and the hepatocellular carcinomas that arise. And we know this in terms of their etiology, the frequency in which they occur. There's lots of statistical and associative studies that show that virus infection is incredibly well associated with both CCA and HCC. And also there's a huge raft of studies that are becoming more prominent where the genetic similarities in terms of gene expression show that they're very similar. Now, hepatitis C is the virus that we're looking at in particular, and it's responsible for around a third of hepatocellular carcinomas and an increasing number of intrahepatic cholangiocarcinomas. Hepatitis C is a unique oncogenic virus. It's an RNA virus, which means it replicates in the cytoplasm. It doesn't enter the nucleus, and it doesn't really encode an obvious, strong, dominant oncogene in the way that something like Epstein-Barr virus does, for example. We also know that hepatitis C causes liver cancers, primary liver cancers, through both indirect and direct methods. And the indirect methods are a consequence of the chronic hepatitis which ensues following hepatitis C virus infection. And the direct effects are a result of gene expression changes induced by this virus, which promote many of the cancer hallmarks that we all know are associated with oncogenesis within the liver. So why are both of these processes important? Well, if you think about the etiology of these cancers, they occur during chronic virus infection, which can take many, many years for tumors to arise in. Obviously, the virus doesn't do this on purpose, and it occurs as a stochastic feature. However, what's different about the liver compared with other organs in our body relates to this chap here, which is Prometheus. And Prometheus, in Greek mythology, was punished by the gods by having his liver pecked out by an eagle for giving fire to man. And every day this would happen, and every day his liver would grow back. And the reason for that is that our liver is uniquely regenerative in nature. Now, our hepatocytes, the main body of the cells in the liver, can regenerate to a partial extent, but by far and away the most prolific means of restoring the cells within our liver in response to damage, and certainly the way that it happens in long-term chronic virus infections, is through stimulation of liver progenitor cells, termed LPCs here. These reside near the bile duct, and they can give rise to both hepatocytes, so liver cells, and cholangiocytes, the cells which line the intrahepatic portions of the bile duct. And this is incredibly important because this so-called ductular reaction is responsible for repairing the vast majority of the damage that ensues upon hepatitis C infection. And so therefore it's this process that we were interested in looking at to see whether this might be perturbed in relation to virus infection and therefore give rise to either a discordant replication of hepatocytes or cholangiocytes with respect to liver cancer. So this schematic here just sort of summarizes the environment in which hepatitis C is infecting the liver and the changes in cellular architecture and milieus that can occur as a result. I've mentioned here these hepatic progenitor cells, and these can give rise to hepatocytes and or cholangiocytes. But of course, for that reason, they can also give rise to carcinomas arising from these cells. Now, interestingly, the liver is incredibly plastic, and in actual fact, these are not one-way processes. These cells can de-differentiate, they can trans-differentiate between the two, because this environment in the liver is unique in terms of its ability to adapt and change in response to injury. But what we're interested in is the idea that this hepatic progenitor cell might give rise to these tumors directly. I'm not going to talk about the mixed phenotype of cholangiomas and Hep C, HCCs today. So our hypothesis was around the fact that hepatitis C might be able to infect these progenitor cells and thereby cause both the indirect inflammatory responses in the liver and the direct carcinogenic effects, and that this might give us a clue as to what might be going on in both forms of cancer. And in support of this idea, before we got started, we knew that you can detect hepatitis C antigen within bile tissue. And because mature bile cells do not have the right receptors for the virus to infect them as mature cells, we know that this has to arise through infection of progenitor cells. And similarly, we can recapitulate these sorts of scenarios in the laboratory. We also know that stem cell-derived models are susceptible to infection. However, one important aspect of hepatitis C therapy nowadays is that we have these new drugs available. And so it was interesting to look at this scenario where surely this is going to diminish the amount of hepatitis C virus positive tumors which ensue. Sadly, this isn't the case. And what we're finding is now that even though people are being cured of their hepatitis C virus infection, they're still at profound risk of developing either HCC or indeed intrahepatic cholangiocarcinoma. And one intriguing aspect of this that's been researched by collaborators of ours as well as in our laboratory is that hepatitis C, although it's an RNA virus replicating in the cytosol, is actually able to profoundly affect the gene expression within hepatic cells after it's been cured of infection using these fantastic antiviral drugs. So we decided that our hypothesis would probably comprise the idea that Hep C is inducing this hit and run type of carcinogenesis, and that the gene expression changes that ensue following this might well be imprinted in a permanent fashion within the cells in the liver, whether they be progenitor cells going on to become a cholangiocyte or indeed a hepatocyte. And that this pro-tumorigenic signature could give us both insight into the processes involved, as well as potentially targets that we could use to try and ameliorate the effects of this virus infection upon cancer causation. So obviously, the first thing we needed to do was prove that liver progenitor cells within the mature liver could be infected by hepatitis C. And that's what we're just very briefly summarizing on this slide here. What we were able to do is to harvest these and grow these mature liver progenitor cells from adult livers available to us at St. James's Hospital in Leeds. And this is what they look like. They're highly pleomorphic, so many different shapes. They have different morphologies and origins, and they grow in different fashions. And I don't have time to go into the ins and outs of culturing these cells yet, or the characterizations that we have. But suffice to say, what we were able to do is label these progenitor cells in green and culture them alongside cells that are highly permissive for hepatitis C virus infection, these HUH7 cells, which I'll come on to in a moment. And when we did that, we were very, very careful to ensure that we could not detect anything aberrant in our non-green labeled cells. But when we're culturing them with our infected cells, we could readily detect these cells that are labeled with a 3 here, that are both green, indicating that they are progenitor cells, and red, indicating that they are infected with hepatitis C. And this, as far as we know, is the first demonstration of hepatitis C being able to infect these sorts of cells from a mature ex vivo liver sample, albeit in a culture dish. But it's at least proof of principle that this phenomenon is achievable within the liver itself. So this is all, but as you might imagine, handling primary cells taken out from somebody's liver is trivial at all. And actually the number of cells that we can manipulate at any one time is limiting. So what we needed was a robust cell culture system that allowed us to study the genetic effects of hepatitis C infection in abundance. So something that we can use as a real workhorse to churn through these sorts of experiments. Now, we're very limited working with hepatitis C because there's really only one cell culture system for this virus, and that's using these human hepatoma 7 cells, HUH7 cells. And these are obviously transformed HCC-derived cell lines. And so they're not really ideal for looking at the precancerous causation type stages of investigating carcinogenic effects. However, what we do know about these cells is that they're not actually one single pure population, and they actually express different amounts of what we might call procarcinogenic factors or cancer stem cell markers within any one single culture. So we took advantage of this fact and separated HUH7 cells based on the expression of a very well-known cancer marker, this protein CD24, which you can see here in green, is expressed in much higher abundance when these cells are actually forming tumors within, in this case, a mouse xenograft. And when we did that, what we were able to derive is these CD24 low level expressing cells here, shown in blue, which were really very bad at forming tumors within our mouse models. And these actually went out for many dozens and hundreds of days without causing tumors with any great incidence in these mouse models. If you compare that with normal HUH7 cells, these come down pretty quickly. So this is showing the growth of tumors in these mice. And if we select for a very high level expression of CD24, these come down extremely quickly indeed. Now, what we're able to do in the laboratory is model this process using HUH7 cells with this low level of CD24 chemically. So we were able to add this chemical called DMSO, and that led to a differentiation process from our model hepatic progenitor cells towards terminally differentiated hepatocytes and cholangiocytes. And you can see that here. This is a picture of these cells using a light microscope, and you can see that from the beginning, they start to develop these characteristics that look much more like hepatocyte cells and cholangiocyte cells. And I'm just marking out here the hepatocyte-like cells in yellow. However, when we did this when hepatitis C virus was present, this process did not happen to anywhere near the same degree or frequency as we saw in our controls, suggesting that the virus is able to inhibit the process of hepatic differentiation within this cell culture model. And this fits with the idea of perturbing the hepatic progenitor cell differentiation process whilst these were trying to restore the liver during infection. We can use various different biochemical markers to check this process, and this is showing our cells at the beginning of these experiments. This is a marker for cells showing that they are proliferating, as you might expect a transformed cell line to do. They're not expressing high levels of a liver marker, this protein albumin. However, when you differentiate them in a normal control aspect, these lose the ability to proliferate and start to turn into hepatocyte-like cells. However, when hepatitis C is present, they continue to proliferate and don't express these differentiation markers. So something the virus is doing during infection is preventing this natural process, albeit induced chemically, in the laboratory. And one clue we had to this is that actually, if you look at these cells as they're differentiating, they look very different whether the virus is present or not. And this is one particular marker here called epcamp. And it's just to highlight really these cells are very structurally different. And this gave us a clue, along with various gene expression trials that we did, that this particular creature here, hippo, might be involved. Now, hippo is a bit of a cartoon notion. Actually, it's named after a cell signaling pathway, which is intrinsically linked to the structure of cells. And this hippo pathway is actually what defines the size and shape of the liver itself in humans and in other creatures. And what hippo does is regulate these two proto-oncogenes, so cancer-causing genes, which enable stemness and delay differentiation in cells, YAP and TAZ. And the hippo pathway itself is linked to the extracellular environment. It runs through a kinase cascade through the cytoplasm, which leads to the phosphorylation of these proto-oncogenes and prevents them entering the nucleus to switch on gene expressions that would make these cells more like stem cells. So we hypothesized that hepatitis C was able to inhibit this signaling pathway and thereby allow YAP and TAZ to enter the nucleus and do their jobs. And we could recapitulate this by adding an inhibitor of this master kinase at the top of the cascade, MST1. And you could use this drug and actually recapitulate the phenotype that we saw when the virus is present by treating control cells with this drug. And this told us that hepatitis C was delaying differentiation through this mechanism. And we're just seeing this in an analysis of protein expression here through Western blots, that like hepatitis C, we're seeing much less expression of this hepatic marker where this drug treatment is present. So we know that this is the pathway that the virus is manipulating. And we can see this because if we look in hepatitis C virus infected cells, this is a viral marker here, and this is the YAP proto-oncogene expressed in these cells, and you can see in the virus-infected cells it's entering the nucleus, whereas it's not in our controls. And again, this is associated with continued proliferation of the cells. So what we wanted to do is get a greater understanding of what was going on in this situation. And what we did was do genomic gene expression analysis of either cells expressing the virus following infection or not, in the presence of various inhibitors that either inhibited hippo pathway signaling, or indeed in the presence of these drugs which abrogate virus replication. And we look using a technique called RNA sequencing to get a global idea of the gene expression within these different experimental conditions. And this just shows us that the conditions were very different. It's something called a principal component analysis. Now, don't be confused by this. What this is showing is the ups and downs of gene expression through different clusterings of the genes that are switched on and off during virus infection. Don't get too worried about this, but basically yellow is up, blue is down. And what we can see if we flip this around 90 degrees and look at it in profile is that many of these genes are switched on during infection, but switched back off again if we get rid of the virus, but some of them aren't. So this fits with the idea that the virus is able to imprint changes into these cells during infection that might cause eventual processes to lead to carcinogenesis. And we saw this by comparing the genes that were changed in virus infection compared with that in cure. This is just a Venn diagram to show there's a huge overlap between these different gene signatures where the virus is either present or has been cured. And what we can see is that there are different transcription factors, so things that switch on genes, and that they activate different pathways and are controlled by different pathways of gene expression. And of course, you can see hippo coming up here in this overlapping signature. So something's going on where the virus is involved to switch these genes on during infection. And if we look at the etiologies of these genes, we can see that they are very much involved in cancer causation, in EGFR signaling, and in hippo signaling. This is just a metabolic signature that the virus induces. This really is indicative of the fact that the virus is able to imprint long-lasting changes in gene expression, which are ultimately going to be pro-carcinogenic. Now, this is a bit of a more detailed picture of the hippo signaling pathway. Please don't be confused by it, but suffice to say, we saw multiple aspects of this pathway being dysregulated during virus infection. And one thing I just wanted to highlight in particular is one example I'm just going to show you of a master transcription factor that's dysregulated by the virus and upregulated during infection, as you can see here. That's actually, we think, behind some of the processes that are perturbing the differentiation signature during infection. And you can see that this protein FOXM1 is a transcription factor, and it's associated with poor prognosis in primary liver cancers. So we can inhibit the activity of this transcription factor using these drugs here. And when we do that, compared with our virus-containing controls, which maintain cellular proliferation, marked out here by this Ki67 protein, in the presence of these drugs, the virus is unable to dysregulate the differentiation process in our experimental systems. So that tells us that FOXM1 is an important tool that the virus uses to dysregulate pro-carcinogenic gene expression. So what about, this is in a mixed system where we have hepatocytes and cholangiocytes. What about in systems that are just tailored towards cholangiocytes? Well, that's what Alex has been focusing on during her PhD. And this is a schematic of hepatic differentiation, again, which we've just been looking at. But what we're trying to do is force this just towards a cholangiocyte pathway. And we can do this using stem cells all the way through here, or we can start at this liver progenitor cell stage that we've been talking about just now. And we have different cell systems that we've been looking at. I'm just going to tell you about two here. One is stem cells themselves, and the other is, again, our H287 CD24 low system that I've just told you about. So Alex has been doing a tremendous amount of work trying to get induced pluripotent stem cells working to generate these three-dimensional models of cholangiocyte-like cells. And this is just showing you how she's managed to achieve this over the course of the last few months, obviously unfortunately interrupted by lockdowns. And what we're able to do now is take these cells at this stage into the laboratory and infect them with hepatitis C, and very much like our H287 systems, see whether this process of differentiation becomes divergent as a result of the virus inducing perturbed gene expression. These are just some more controls for those who have a bit more specific knowledge about these sorts of systems, showing that what we're doing in these three-dimensional cholangiocyte-like structures is we're switching on specific cholangiocyte gene expression and morphologies, and we're switching off hepatocyte-related gene expression. And we have very little proliferation in these terminally differentiated models. However, again, we need a workhorse for these systems that we're looking at in terms of cholangiocyte differentiation. And Alex was able to generate a modified version of our systems that we've just been talking about, incorporating the fact that sodium taurocholate and TGF-beta can profoundly affect the differentiation processes within the liver, and specifically drive things towards biliary differentiation. Again, Hippo is involved because we're having to use DMSO to activate the MST1 kinase. And what this is showing you here is that the morphology of our cholangiocyte-derived cells here in the HPH system are very different to the hepatocyte. They're not cells in cell cycle and proliferating, so again, they're becoming differentiated. And we're switching on cholangiocyte genes, but not hepatocyte genes. So Alex has also been doing a of genomics analysis, and I'm not going to go into details on what these are, but suffice to say what we're trying to do is relate our RNA sequencing data to real-world data from patients and other studies in mice, for example, to see if we can try and identify some common factors between what the virus is doing in various systems to underpin what we're understanding about the differentiation process, and also to really highlight targets that we can use therapeutically to prevent the causation of cancer following cure of this virus. Now, these different studies overlapping with our data, and I've only shown you these really because they make pretty diagrams. These are actually Venn diagrams. But in the middle of all of these diagrams are a list of genes that are common to the virus in different situations in different cancer-causing models. And what we noticed was that these two proteins here seem to be coming out in many different screens that we undertake during these genetic analyses. And just to explain a little bit about this TMEM45 is a transmembrane protein. It's involved in cellular differentiation paths, you might expect. It activates things like resignaling, and again, is associated with a relatively poor prognosis in primary liver cancers. Very quickly, this neuregulin protein, it can be secreted. It's an EGFR-like ligand. It activates EGFR receptor processing, and it also activates Hippo signal in hepatic cells as well. And the reason we're interested in this protein neuregulin is A, because it's secreted, so it's therefore targetable by things that will block secreted factors, but also because we know that the conditioned supernatant from our virus-infected cells are themselves capable of inhibiting the differentiation processes in our model systems. And that's by activating the Hippo signaling pathway, increasing the expression of things like TAS and YAP. So again, these secreted combined with infected cells factors could present really excellent targets for us to target to prevent the onset of carcinomas within infected patients. So I'll just leave it there to summarize that we are using hepatitis model of a pro-oncogenic driver during the differentiation process within the liver that's derived from liver progenitor cells. And we believe that hepatitis C is perturbing Hippo pathways amongst others to induce a de-differentiated state in LPs, in liver progenitor cells, which ultimately dysregulate the production of cholangiocytes and hepatocytes and can eventually lead to cancer causation. And if we can identify at-risk gene signatures within these sorts of systems, then ideally we'll be able to take these forward into therapies in the future. And I've shown you a few examples of those. So again, this is a huge group effort, mainly driven by Alex, Avi, and Matt, but also in huge collaboration with Adel, Andrew, and others in Leeds and elsewhere. And I'd like to sincerely thank AMMF for enabling these studies to occur, and also the Medical Research Council for previous funding. Thanks very much.